time is really passing fast. So it's over 19 years already. And as Pedek, I don't have a direct ancestry line with Sue, but we have shared many moments here at campus for seminars or events or congresses or other meetings or visits of Sue and her group to Highline or many other events in the US or internationally. One thing that I have always admired from Sue through the years in her presentations is that she can put these complex processes for immunology and immunogenetics in a way that you think that you understand them. So it's great. But I think in our relationship has been mainly one direction. I'm learning from her all the time and enjoying all the experience that she carries. But in the personal level, I couldn't agree more with some of the comments previously in terms of her empathy, the unselfish willingness to share knowledge, and her very good sense of humor. I don't know how many times I have seen Sue in meetings, but within seconds or within a minute, we will be saying something that will make us laugh. It's a joke. Usually, it's related with food, and so I will always keep memories of Sue of that, the kindness, the sense of humor, and the beautiful smile that she always has. So thank you so much, Sue, for that. So you will maybe wonder what the symposium at Sue has to do with cacao plant or chocolate. And I have an anecdote. I have nice memories of this symposium, of this animal health symposium. I learned a lot of things in that meeting. I met a lot of great people, but there was something for free that will stay with me for the rest of my life. You know, I grew up in the tropics. My father was a coffee grower and trader, and he used to have cacao trees in our farm, and he will process very rudimentarily, very simply, just for self-consumption and for selling the local market. So my mother has an abundance of cacao paste at home, and she made it was our breakfast every day, and was in foods and treats all day. So I became addicted to cacao and chocolate. So, but in the wildest imagination, I will imagine that something like a chocolate fountain existed until I came to in 2005, and I said, "Wow, this is an eureka moment for me." In the dinner of that event, Sue presented us with this, not only a chocolate fountain, but strawberries, which I love, all kind of berries. So can you imagine to have two of your favorite foods at Livingston in front of you? And I don't know how many of those things I ate, but I'm sure that I ate more than my fair share. I probably ate enough for Peteg and Janet over there too. So, but let's talk about a different kind of food. If this helps me. Maybe the battery is gone. The presentation. Yeah, it's frozen. It's not advancing. Okay. So let's talk about broiler. And I think Jack suggested when he kindly invited us to come and participate in an event, he suggested that we talk a little bit about the future. But it's difficult to talk about the future without knowing how we got where we are today. And I think Petek did a great job in introducing the origins of this and the history in the life side. I'm not going to talk much about genetic improvement because time, basically. If I'm going to talk a little bit of the projecting, it's difficult to define this into much detail. A little bit of the peak of the future and into the future, and this doesn't intend to be comprehensive. I mean, is my perspective is probably biased towards what Cobb is doing right now, and we are envisioning to do. Maybe there are other visions for the future. So to talk a little bit about history, I borrowed this all graphs from an old presentation from Cobb and just modified it to extend it to include genomic selection and to capture 1 century, 100 years. And Petek mentioned when the times when this poultry was produced with dual purpose, and how, you know, the chicken has this dilemma, to lay or to grow. You cannot ask for those two at the same time if you want to be efficient. So they started the the selection and diversification of lines specialized for eggs, and then the layer industry developed, and the ones for meat, and then the broiler industry became a reality. And so mass selection for growth really pushed the progress in broilers. And about the same time here in our state, Loesch and later Hazel developed the selection index, which was spread, and it was really gave a framework and tool for selection. It is difficult to go through many of these other aspects for the sake of time, so I only highlight the ones that I think are really crucial and important for us here today. I think during this time, Henderson was busy developing, and many others, and variance methods for estimation of variance components, the mixed model equations. But it was not until efficient algorithms were available and computer power that the animal models of group progress really had an impact in the industry, starting with dairy, and of course it spread to all the other species. Later then came the time of the era of the DNA markers, molecular markers, and we all remember those papers with marker-assisted selection and QTLs, and that evolved quite quickly and rapidly thanks to the development of the human genome into having dense chips or markers that could be combined, and then the idea of the methodology of genomic selection. And I think in the last 15 years, both the industry and the academia research has been busy not only in developing the best ways to implement genomics, but allowing this technology to be applicable because of the reduction in cost of the genotyping. And I remember, I think this reminds me the times in which Evigen started their genomics projects, and they established this genomic advisory committee in which Sue and Jack were always present, and we have these interesting meetings, but also Rohan and Fernando and later Dorian Gary. So really, we have gone through quite a lot of generations of genetic improvement that has been enabled by genomics. And you probably have seen many iterations of this graph. This is an infograph that is available in the website of the National Chicken Council. And those studies that compare the Athens Canadian random breed with the modern broilers. And what really impacts me of this is that the difference in the old chick body weight is about 30%, but if you compare at 28 or 56 days, it's for over 400%. So really the progress has been tremendous in terms of the ability of those broiler chickens to grow rapidly and became really meat machines. And I think everybody in this room is familiar with this pyramid. And so, but this allowed me to talk a little bit of the present and the future. We have the intense selections in these elite or pedigree populations that we will continue selecting intensively with genomics. And I think Joseph gave us today a real class on all the possibilities that genomics can offer. And that will continue, of course. And then there is the pipeline, the commercial pipeline that has two main purposes. One is to multiply the populations to a level that can satisfy the supply needs. But the other thing is to exploit some of these benefits that come with the crossbreeding, heterosis, line combining ability, and complementarity. But part of the future then, besides optimizing this pyramid at the apex of it on intense selection of pedigree lines, is to utilize the pipeline for research. And that's what we have done in Cobb in last year. We established what is called the proving grounds. It's basically a replicate of the entire pipeline, the crossbreeding, but for the unique intention of research. And I will explain that in more detail a little later. So to finish with the history, there is no doubt that after 2010s, with the reduction of genotyping cost, all the industries started implementing genomics, and I'm not going to talk about other species for instance, dairy or swine, because I think with Joseph's presentation this morning is more than clear. But I would like to talk a little bit about the journey for the poultry, which really started in 2004 with the publication of the first draft sequence of the chicken genome, the first livestock species or agricultural animal species that were sequenced. And Sue has a very important role to play in this job, this work that I think was really a cornerstone for the implementation of genomics in poultry. After that, then different kind of snitches were developed, each time larger, bigger and bigger chips. And in the last 15 years, all the companies and the industry in general, but the research and academia, we have access to these SNP chips, medium or large density SNP chips to implement genomics. I borrowed this slide from Anya, which remind me to explain how this genomic started, at least in Highline in 2009, with one experimental line, and how there were applications for this USDA grant that were given to the group of Cobb and Hendricks. And then within Highline, we said, well, what we do now? So we needed to fund this at the time when I was in Highline. And Jack helped Anya in designing this experiment in the way that we could do it solo with our own resources. And even though it was a small population size, in 3 years we could demonstrate that the advantages of genomics. And I think it convinced us at that time that there was the way to go in the future. So I think it was the starting point of the implementation. And Petek showed that in 2012 that the first commercial line was included in genomics, and at the same time, Evgen announced the implementation of genomic selection. So I think that genomic selection now is a methodology that is embedded in the breeding programs, is used in routine, and I think the industry now have focused in investing resources and talent in terms of phenotyping. I think if we learned something during the genomic implementation era, is to realize that the phenotyping is king. I mean, genomic tools doesn't work if you don't have the right phenotypes and the continued evolving into the capturing phenotypic information. So precision agriculture or precision farming open all of these portfolio of technologies that can be applied to phenotype more efficiently animals. And I think that's really the future in broiler genetics. And this started maybe with crops because they had early access to this satellite or GPS, and later drone facilitated aerial technologies to monitor entire plots in large scale for macro environments, and then also the micro environment level at the plant level. And this has been adopted in livestock, for instance, in swines, computer vision to improve traits related to welfare, especially walking ability. And importantly, there are many examples in the literature. I have these two examples of research that has been funded by Cobb. On the top is a Dutch government consortium that is in collaboration with the University of Wageningen in the Netherlands, that has used a combination of RFIDs and computer vision to study behavior, feeding behavior, drinking water behavior, but also outside of the pens in platform studying walking ability and gait score. And in the bottom is a research that is funded to the University of Georgia, the similar technology, and they are not only focusing in tracking individual animals and then also working on gait scoring and walking ability, but in ways to develop computer vision systems that are portable, affordable, and can be scaled up for large scale implementation. So if a company wants to be best in class in terms of R&D implementation of these technologies, it has some challenges. It has a lot of opportunities, but also it has challenges. It's not a low cost. especially if you want not to do it only in a research setting with a small group of animals, but you want to implement it in the breeding programs, where you have to extend it to multiple farms, multiple populations, and thousands of individuals if you want to really use this information in the breeding program. So it's a cost associated with talents, hardware, software, and somebody was mentioning storage, because this is large data sets that are accumulated when you scale this up. And there is competition, not only within agriculture, plant, animal agriculture, but also basically all the economic activities of society are using this kind of technologies. Other thing that is important is that you need to really focus what are the traits that you are going to emphasize with this technology, because there are so many possibilities, and there are so many possibilities that you have the problems where I put my resources and my talents. Fortunately, there are a lot of groups around the world working on this. And so the approach that Cobb is taking is to fund research partnership with different institutions at the global level in what is called the Cobb Research Initiative. And I invite you and people that you know that is interested to apply for this funding. So the Cobb Research Initiative is basically to promote collaborations, direct collaborations that are funded by Cobb, and is to develop technologies that can be implemented in the breeding programs to improve collection and data, or methods to for prediction or estimations. So right now there are 9 projects underway in this CRI. It started in 2023, so 2 years. This year alone, we had 42 applications, and these are the 9 areas of focus. And these columns represents the number of applications that we're receiving each one of those areas. So you can see that AI and precision farming was number 1 with 11 applications. Then broiler livability, and if you add to broiler livability microbiology, because it's related with many pathogens that has to do with the mortality in broiler, then is a very competitive area, but also in genomics. So what the committee decided to do for this year is to refocus the priorities in terms of artificial intelligence, data system, robotics, and automation, and application of biotechnology. And mainly application of biotechnology applications related with the traits associated with the disease and livability of broilers. So just to give you a flavor of some of these projects that are underway and already funded, I already mentioned this project with the University of Georgia, looking at the individual tracking and walking ability of broilers in small settings, but looking at ways to scale this up in real applications. Oh, it's not moving again. Oh, here. I think it's because it has a video that is embedded, so the same problem that don't have had. So this is a project in collaboration with the University of Tennessee and the University of Maryland, and it's the same computer vision idea, but in this case, not in small pens, but in large breeder houses, commercial breeder houses. And there is this video that you cannot see, but it's basically focusing in mating behavior. We know fertility is a very important problem in the industry right now. So trying to associate mating behavior and fertility of males in those commercial scales breeding houses. And this data also allows us to capture more information in terms of nesting behavior, incidence of floor eggs, and other behavioral traits. But the main focus is on mating behavior and effective mating in broilers. Another technology that I think is really a game changer in broiler breeders and broiler genetics is the implementation of CT scanning. It started a few years ago as a proof of concept, but now it's fully implemented across pedigree in all the pedigree farms for all experimental and commercial pedigree lines. And the main advantage of the CT is that is performed in live animals. It means that you can measure these traits in selection candidates. In the past, many of the traits related to body composition, tissue distribution, meat quality, yield, breast milk yield, and also associated with the skeletal integrity and leg health, especially things associated related to traits like tibial chondroplasia or femoral hair necrosis, require sacrificing birds, sending it to processing plants or doing infant dissections, and the animals be very used to do this kind of process in the farms. Now with CT, you can basically have live bird cuts in candidate of selection without the need to use sieves for those traits. And so traits like this related to muscle yield and body composition and skeletal integrity are completely, fully implemented across pedigree. But there are other traits that are under exploration right now that are related to metabolic health and gonadal development. And the beauty of the CT scanning is once you take these scans for birds, you have this collection of X-rays that you store. It's a very large amount of data, but you can revisit those scans as many times as you need in terms of annotate or curate those scans for any other traits that you want in the future. So this data stays in storage, and you can come back to this data as many times as you need. So there's another CRI project with the Georgia Tech that focus specifically in the application of CT scanning for cardiovascular health, especially associated with the metabolic syndrome in broiler ascites. And looking at features related to heart shape, volume, density, etc., traits that can be associated with those metabolic conditions. And as I said before, CT and these technologies are applied in all the pedigree farms in the system. And then in the Delmarva region, there is what we call proving grounds. And I mentioned it before. So proving grounds is a complete replication of the pipeline in a concentrated area. So there are GGPs, GPs, and parents in the same geographical area. So it's easy to mobilize this genetic material and then really scale up the possibility of evaluating the current commercial products and the emerging crosses and alternative new products. So, and the basic idea behind is to really accelerate the production of new products, to diversify the portfolio of products, or to optimize the ones that are currently available. So basically what happened is all the lines from all the pedigree lines, experimental and commercial pedigree lines, are sent to Delmarva as GGPs, and then all these crosses that replicate the latest generation of the commercial product today, but also of the alternative crosses are compared in the same environments at large scale. And when I say large scale, is when this system is working properly, there will be the ability to test about 100 times more parent stock and about 400 times more broilers. So the time of small trials that require many replicas across time, that take a lot of time to decide if a specific cross or new product has potential in the market, will be accelerated because they will be produced with a much higher speed and a much larger volume. So I think this really will transform the way the products are developed and maintained in the system. So I want to emphasize that I think, talking about the future, I think the future is mainly in the area of infusing technology and best practices. I have an example here of some technologies that are fully implemented in the pedigree system today. I already mentioned CT, but there are others. And things that are in common of these technologies, the first one is that all of them are very animal friendly. They don't require sacrificing animals or working on sheep post-mortem. They are very relevant for phenotypes that are important in the industry today. They produce high throughput of data, large amounts of data that can be exploited in the breeding program, and many of them are real time. So besides the CT scanning, the pulsometer to measure oxygen saturation has been a very good marker for cardiopulmonary function and activity in broilers, and we are implementing it across all the pedigrees and experimental lines. Real-time feed conversion has been extended now, even in experimental farms. And I think a technology that really will mark the difference in the future is near infrared. In the past, I have seen application of near infrared, for instance, for meat quality and meat composition in fillets, in post-mortem. But microRNA has a versatility that can be used in live animals and predict many of these traits in candidates of selection. So I think this is the real future of the breeding program. And there is another area in which I want to bring an example that is not in poultry, it's in swine. But and I mentioned in a question this morning, is related to the application of other genetic technologies, but especially the gene editing. It's an area that we really need to continue investing and working on research, because I think this will be really a savior in the future for this industry. PAC Genus not only develop all the basic science to introduce gene editing to produce pigs that are resistant to PRRS virus, but also went all the way to explore the marketing possibilities, went to the old regulatory process, and then study very carefully the preparedness to go to market with this technology. And what they said and they concluded is a progressive process, and it takes a lot of time. They speculate in this publication that it will take about 10 years from populations that are completely susceptible or naive to have a commercial population that are completely resistant to the virus. So if we need to, if we need and want to implement these technologies in the future, we need to invest in the basic research today. And I know that all the breeding companies in poultry are investing in this through collaboration with research institutions. And it's something that we have to encourage and continue. So just to finish, I think that I always say that in breeding and genetics, the only constant is change. And I think the future will continue bringing change. And this change is in the form of short-term, medium-term, and long-term. In the short term, we need to keep growing and intensifying the implementations of new technology. This is related with the AI, digital phenotyping, etc. And then partner with researchers around the world to fine-tune and find which one of these specific technologies can be implemented and in which areas of the breeding program, and then accelerating the product development in this idea of the proving grounds. But I think to finish, and this is my last slide, I think I'm okay. Today is about Sue. So when I received the invitation, I went to the Iowa State official website and I downloaded Sue's CV, and I was really impressed. And I said, I need to summarize this somehow. But instead of reinventing the wheel, I cheated and I went to ResearchGate and see those statistics. And if you see the research interest score of Sue is close to 6,000 with more than 70,000 reads, 11,000 citations, more than 300 recommendations, and 533 research items. And this is really incredible. Not only that is incredible, if you see the ranking according to ResearchGate, she's in the one top percentile in the entire research community. That means that she's competing with other areas of science that we know that have a much smaller minimum publication items and also much shorter time frame of research. So my final question was Sue slowing down now that she knows that retiring? And what I found in ResearchGate is that in the last 6 years, she has kept about more than 600 or more citations per year, and she has produced 128 research items in this time. So this is incredible. Thank you so much, Sue, for your legacy, and especially for the human being that you are. It's really has been a really great honor for me to able to share, and I hope to see you many more times, and keep your smile. Thank you so much. I don't know if there is time for questions or.
Maybe one quick question as our next speaker comes forward, if somebody has one. Oh, I see one in the back.
Yes.
Yeah, we have to run.
So, Jesus, what can you not do today that you wish would cost 100 times less and have a 100-fold scale up?
Wow. There's a question of a wish. I don't know if I can grasp this question. It's a very good question. What I can say is that we need to keep changing and adapting, and there are so many technologies out there that are available that we can scale up. You just need to put together the right talents and the right resources, and that for me that's a big challenge. I can have a list of 10 things that I want to do tomorrow that I need to find exactly fine-tune how to implement them. And the idea of this presentation was to kind of present some of the things that we are doing that we think that are taking us to the right direction. I think in broiler breeders, especially, having technologies that allow you to measure the traits, the relevant traits in candidate selection, that's key. Because having this system of relying on SIP information that requires sacrificing birds in a limited amount is not only a very expensive proposition, but it's also not the best if you have alternatives like CT that allow or an NRI that allow you to take those traits in the candidates selection.
Thank you.
Thank you.